Hi, I'm Kiani, and I'm going to talk about my final year honours project, which was looking at the molecular mechanisms that underlie the activity regulation of the tumour suppressor protein deleted in liver cancer 1, or DLC1. So, it has been previously reported that DLC1 undergoes an autoinhibitory interaction that greatly reduces its function, and this is what I was investigating using a structural biology technique known as NMR. In order to investigate this interaction, various fragments of the protein DLC1 were cloned into the bacteria E. coli. The bacteria are then grown in flasks and induced with IPTG, which is a chemical that causes them to express the desired protein. All the proteins are then extracted from the E. coli cells. The protein that we're interested in can then be purified away from all of the others using a process called affinity chromatography. The protein that we want has a tag on it that will enable it to bind to a nickel-containing column, whereas all of the other proteins will not bind to the column. The purified proteins are then made into samples for NMR analysis. The samples are pipetted into thin glass tubes, which can be put directly into the spectrometers. NMR is a technique that uses large magnets to generate strong magnetic fields. When a sample is placed in the magnetic field, its atomic nuclei can absorb and re-emit electromagnetic radiation. The re-emitted radiation gives us information about the position and environment that the atomic nuclei are in, which is related to the structure of the protein. Data from the experiment is displayed as a series of peaks. We obtained one spectrum from the catalytic domain of DLC1, and another spectrum of the catalytic domain plus the part of the protein implicated in the autoinhibitory interaction. Then we could directly compare the two spectra. When we compared the two spectra, we could see that some of the peaks were broader, and some of the peaks had shifted in position when the sample contained both proteins compared to the sample only containing one. This is indicative of binding, and therefore supports the proposal that there is an autoinhibitory interaction, regulating the activity of DLC1. However, further investigation needs to be done to determine exactly which amino acids of each part of the protein are interacting with each other. But hopefully, this information will contribute to the understanding of how this important tumour suppressor protein works.